Hello, good morning, good afternoon, good evening, wherever you are. Welcome to another session in this gene therapy course. And today's topic is the DNA design, the gene design. We are going to learn how to design the OSK genes, these OSK genes that we have here, the therapy to rejuvenate the DNA. Okay? In the previous video, you learned how to design the plasmid DNA that carries the OS genes. So in this video, we are going to learn how to design the OS genes, and it's very easy. Okay, the OS genes are structured kind of in layers. Okay, and the first layer that involves that wraps the whole thing are the restriction site 1 and the restriction site 2. You see here, I have marked them with a label number 1, with the label number 1 here and here. So the restriction site 1 and 2, these sequences are responsible for inserting, for cloning the OS genes, everything that you see here inside the plasmid, thanks to the multi-cloning site. Okay, so the restriction site 1 and 2 is responsible for inserting the OS genes inside the plasmid. Then what we have? What we have here are the ITR sequences. This one here and this one here, the 5' and the 3'. And again, it's kind of a layer. It's wrapping, it's involving, it's escorting the OS genes. And these sequences are kind of labels. With these sequences, the mammalian cells are able to recognize what is inside. So the HEC293 cells start copying many times only this sequence from the ITR 5' to the 3' ITR. So they start copying many times this sequence that we have here. And after copying, this label also serves as encapsulation label. So these kind of labels are indicating to the cell that they have to encapsulate the copies inside the adeno-associated virus. Okay, so these sequences, the ITR sequences, are indicating to the cell first, copy many times this, and afterwards, after copying many times it, they have to encapsulate this sequence inside the adeno-associated virus. Yeah, what we have here are the genes. We have a first gene that controls the second one. So this first gene receives a signal, okay, and the protein that is generating thanks to this gene interact with the signal and lead the production of the second gene and the or cassette of genes. And the second cassette of genes that we have here are the OS genes, the genes necessary for rejuvenating our DNA. This is the therapy. Okay, the OS genes that is controlled by the preceding gene that we have here. Okay, so let's dive into it. Let's delve into it. Okay, let's get more details. Let's learn a little bit more about this exciting topic. Let's go for it. Okay, so the first thing that we need to do to design our OS genes is selecting the restriction site 1 and the restriction site 2. We have here the plasmid, the multi-cloning site, okay, and we select 2 restriction sites within the multi-cloning site. Okay, so we will select the restriction site 1 and the restriction site 2. After selecting both of them that are different, we will check that the OS genes don't have inside these restrictions site, that they are free from these sequences. Otherwise, they would be cut, okay? And this would be not nice, okay? So after selecting the restriction site 1 and 2, we copy paste at the beginning of the OS genes the restriction site 1, and we will copy paste the restriction site 2 after the OS genes. That is all, okay? And thanks to this restriction site, we will be able to insert this inside the plasmid, okay? This is the plasmid, and thanks to the restriction site 1 and 2, we will use enzymes that will recognize specifically these sequences, and they will cut here, plop, and here, plop, it will generate a plasmid free from this sequence, and afterwards we will add the OS genes, okay, the OS genes that carry the restriction site 1 and 2, and these extremes are very sticky, so they will get here thanks to the restriction site 1, and will get here thanks to the restriction site 2. They will be bound thanks to these sticky streams, because both of them have been cut with the same restriction size, right? So that's all, my friends. Just copy paste the restriction site 1 at the beginning and the restriction site 2 at the end of the genes. Go for the second layer of information, which are the ITR sequences. Okay, the ITR sequences. Remember, the restriction site 1 and 2 help us to clone this inside the plasmid. And now the ITR serve another purpose. The ITR sequences are kind of labels, okay? They label what they have inside, and what they have inside is the cargo, the genes, the OS genes. These labels, the ITR labels, indicate to the cell, to the HEC296 cells, to mammalian cells, that they have to amplify this sequence. They have to make many copies of this sequence. So these labels indicate to the mammalian cells that they have to produce many copies of what is inside. And these labels also are responsible for the following thing. They are saying to the cell, okay, after making many copies, what you have to do is encapsulating the copies inside the adeno-associated virus. Okay, they have to encapsulate the information inside the adeno-associated virus. Okay, so what are we going to do? Okay, we copy paste the 5' ITR after the restriction site 1 and before the cassette of genes. And then what do? Is copy pasting the 3' ITR after the genes and before the restriction site. Okay, that is this is how it should look like. Okay, so very easy. The labels that are responsible for making many copies of the OS genes and encapsulated them inside the adeno-associated virus. And the way to do it is copy pasting the 5' ITR before the OS genes and the 3' ITR after the OS genes. Very easy. Let's go for the next piece of information. Okay, so let's go for this information. Let's start understanding a little bit about the OS genes and how they are designed. The first thing that we have to know is that in between the ITR sequences, we only can have 4.5 kilobases, so it's the limit. So everything that we design has to fit in 4.5 kilobases. Okay, so we are a little bit limited. Sometimes we will have to shorten certain sequences or using small elements to fit into the 4.5 kilobases. Okay, this is the limit of the cargo that the virus can carry. Okay. Okay, so in this slide we're going to see the first cassette of genes. This cassette of genes produce a protein that is called RTTA4. Okay, these genes are the regulatory genes. These genes control the production or expression of the second cassette of genes, that are the OS genes, the treatments. So these first genes that are regulatory genes produce a protein that is called transactivator protein, in this case RTTA4. RTTA4 is a protein that senses, that feels the presence of the oxycycline. So we give to the patient the oxycycline, and once there is the oxycycline in the media, the protein binds the oxycycline, and once it is bound, is active, is able to interact with the promoter, with the promoter of the second cassette of genes, the OS genes. So once it detects the presence of the oxycycline, it's activated and it starts interacting with the promoter of the second cassette of genes, and the transcription of the second cassette of gene can happen. Okay, so that's why it's called a regulatory gene. And as all the genes they are constituted in the following fashion. Okay, they have elements for transcription first. They have elements for transcriptions, which are the promoter and the poly-A sequences. Okay, the promoter indicates to the RNA polymerase that it has to start transcribing. In other words, producing mRNA. Transcription is the production of mRNA, and the poly-A sequence indicates to the RNA polymerase that it has to stop producing mRNA and adding a tail of poly-A. Okay, as you can see here, you see the virus, right? The virus gets inside the nucleus, the capsid is dissolved, it releases the cargo, the genes, and here you have the first genes that are the genes of the regulatory element. Afterwards, they are translated, produce a protein, and this protein controls the expression of the OS genes. Okay. So as I have said, we have first a promoter. In this case, a minimal CMV, cytomegalovirus promoter. It's very small. Why? Because we are limited in the size of this construct. Okay, and then we have the poly-A. So these both sequences are controlling the transcription, the production of the mRNA. And inside these sequences, what we have is information necessary for translation. The translation is the production of proteins. So we have here the template, which is the mRNA produced thanks to the RNA polymerase. Then this mRNA is exported to the cytosol, and in the cytosol it binds ribosomes that will produce proteins. Okay, they will synthesize proteins. What is the information necessary for producing proteins? Okay, we need information for starting the translation. The necessary information for the translations are two sequences, the Cossack sequence and the start codon. Sometimes the start codon is inside the Cossack sequence, and sometimes the start codon belongs to the genes. Okay, so for initiating the translation, we have the Cossack sequence and the start codon. Both of them are necessary for starting the translation. Then we have the genes, the regulatory element genes, and then we have the stop codon. This is easy, right? The stop codon indicates to the ribosome that it has to stop translating. Okay, so let's summarize this. As in the majority of the cases, the design of these genes are as follows. As always, we have information necessary for transcription, for production of mRNA, which is the promoter and the poly-A. The promoter indicating the initiation of the transcription, the poly-A indicating the ending of the transcription. And inside both sequences, we have information necessary for translation. That is to say, the production of proteins. For starting the translation, we have the Cossack sequence and the start codon, and for finishing the translation, we have the stop codon. And in between the start codon and the stop codon, we have the information that we have the sequence of the genes, of the regulatory genes. Okay? Remember, these genes are necessary for producing this protein that controls the expression of the second cassette of genes. Being the expression, I repeat, the process of transcription and translation. That is to say, the production of mRNA and the production of proteins thanks to the mRNA. Okay? And now we are going to dive into the most interesting topic, the design of the OS genes. These are, in fact, the real treatment, the OS genes that are here. This is information that I have told you is, well, we have designed this because we want a tight control over the treatment, and the way to have a tight control is introducing genes that control the production, that control the expression of the OS genes. Okay, so let's go now to learn a little bit about the OS genes. Before going to the design of the OS genes, I have to tell you that this is very easy. The editing this DNA is easy. Okay, you just have to copy paste first the promoter after the ITR, then you have to copy paste the Cossack sequence, then the sequence of the genes, and afterwards the sequence of the poly-A. So it's very easy. Okay, so now let's go for the design of the OS genes that we have here, the real treatment. Oh yeah, finally, here we are. Okay, understanding the OS genes. Yeah, the OS genes that carry the three proteins. The three proteins that produce the magic, the three proteins that rejuvenate our DNA. Yeah, that help the patients to live a healthy and long life. Okay? Oh yeah. And okay, so let's see them. Okay. Okay, these genes are produced in an mRNA that is called polycistronic. Okay, so a polycistronic mRNA is a mRNA that carries more than one gene. That is to say that will produce more than one protein. So the design of this gene is similar to the previous one, but it includes a couple of sequences that will help it to produce different proteins. Okay, with the same mRNA, thanks to these specific sequences. Thanks to these specific sequences, these proteins will be produced separately using the same mRNA. Okay? Okay, so the first thing that we need to express, remember, expressing means transcription and translation. Transcription is production of mRNA, and translation is production of proteins. Okay? So, okay, the information necessary for transcription. As you have seen in the previous gene, we have a promoter and we have a poly-A sequence. The promoter indicates to the RNA polymerase that it has to start the transcription. It has to start producing the mRNA. And the poly-A indicates to the RNA polymerase that it has to stop. Okay, so thanks to the promoter, the transcription will start, and thanks to the poly-A, it will stop. And the first thing that we see here, okay, is a promoter that is called TREP promoter. It's a inducible promoter. So it will only start the transcription once something happens. What has to happen here? It needs something to be bound, okay, to it, which is the transactivator protein that we saw previously. Okay, so in this promoter we have a regulatory element that binds the transactivator, and once it binds the transactivator, it starts uh, transcribing, producing the mRNA, and once it sees the poly-A, it stops. Okay, so in order to have a tight control over the production of the OS proteins, we need a inducible promoter. Okay, a promoter that is preceded by a regulatory element. Okay, so this is the promoter. Remember, for transcription, promoter and poly-A, and this promoter is a little bit special. It has the regulatory element, so it's inducible. And in between the sequences necessary for transcription, we have the sequences necessary for the translation. That is to say, for the production of the protein. Okay, so let's go for it. Things important to know here. In order to start the translation, we need the Cossack sequence, and sometimes the Cossack carries the start codon. Okay, so for starting the translation, we need the Cossack and the start codon. And the start codon can be within the Cossack sequence or at the beginning of the first gene, the gene of OCT4. Okay, and we will have a stop codon that will be here at the end of the sequence before the poly-A. Okay, so for translation, for initiating the translation, we have the Cossack sequence and the start codon. And the end of the translation will be the stop codon, which is before the poly-A and after the KLF4. Okay, so, okay, we have started the translation, okay, thanks to the Cossack sequence and the start codon, and it starts translating. It starts translating the sequence that is the sequence of OCT4. It starts translating, producing the protein, and hop, you see here, we have a sequence that is called P2A. That sequence that makes is that produce a gap in between this sequence and the following one. So remember, in the whole construct, you only need a start codon and a stop codon. And after the OCT4, we don't need a stop codon because the translation has to carry on. So the ribosome, once it sees the P2A sequence, it will produce a gap so that the next gene will be translated independently. Okay, so we have here the ribosome start producing the protein, the OCT4, it sees the P2A sequence, it produces a gap, and it carry on translating. It carry on translating the sequence of the SOX2, the genes of the SOX2. So it will produce the SOX2 protein, and you see here we have another sequence that is very similar to the P2A. We can, in fact, we can use the same sequence, but for learning purposes, I have used the two different ones. Okay, you can find in the literature different sequences for producing gaps, for expressing multicistronic protein, for producing many copies in a multicistronic fashion. Okay. So, okay, let's remember it a little bit. It starts here the translation in the Cossack using the start codon. It starts producing the OCT4 protein. It sees the P2A. It produces a gap and it starts translating independently the SOX2. It produces the SOX2 and it sees the sequence that is called T2A. In here, the ribosome produces another gap and it starts producing again the protein that is called KLF4, and it produces it independently. The ribosome translated this sequence, produces the protein, and it stops in the stop codon that will be at the end of the KLF4. So let's summarize it a little bit. For transcription, we need a promoter. This promoter is inducible. It requires a Steckner signal, and for finishing the transcription, it requires the poly-A sequence. These two sequences are necessary for transcription. And then we have information necessary for translation. That is all this information from the Cossack sequence up to the stop codon. And what we have is the Cossack sequence and the start codon initiating the translation and the sequences of the different genes. And in between the different genes, we have these sequences that are called P2A and T2A that produce gaps. That is to say, that let us to produce proteins independently in a multicistronic fashion. So what we have to do is copy pasting first this promoter, then the Cossack sequence, then we copy paste the first gene, the OCT4 gene, then we copy paste the P2A sequence, then we copy paste the SOX2 sequence, then we copy paste the T2A sequence. Afterwards the KLF4, and after the KLF4 the stop codon, and after the stop codon the poly-A sequence. Okay, a sequence of different copy paste. So a lot of copy paste, but this is the structure. So I hope you have enjoyed this session. This session that is awesome. The design of the OSK genes finally. Hope you have enjoyed. Have a good day. Bye bye. Bye bye bye. No bye 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 because we have a nice summary here before saying bye bye. This is a nice summary what we have just seen. Okay, let's go for it. Let's remember what we have just seen. Okay, in the design of this DNA, we have first the restriction site 1 and the restriction site 2. Okay, these two sequences are necessary for cloning. That is to insert the DNA inside the plasmid. Okay, then what we have is the ITR sequences. Okay, this ITR sequence in 5' and the one in the 3' in the 3'. These sequences are necessary for making many copies of these and the ITR sequences, and then encapsulating this DNA inside the adeno-associated virus. Okay, and afterwards, what we have is the first cassette of gene controlling the second cassette of gene and the second cassette of gene. Okay, of course. So let's see the regulatory gene, the first cassette of gene that control the OS expression. It has transcription elements, that is the CMV promoter and the poly-A, and then it has translation information, that is the Cossack sequence, the start codon, the RTTA4 sequence, and the stop codon. And then we have the second set of genes, the OS genes, okay? This is the real treatment. We have again exactly the same structure. We have elements for transcription, that is the TRE promoter and the poly-A. The poly-A. Okay, these two sequences are necessary for transcribing. And then we have information for translation. This is the Cossack sequence, start codon that starts the, so these two sequences are necessary for start the translation. Then we have the OC4 sequence gene. Then we have this sequence that produces a gap. Then we have the SOX2 gene that produces this protein. Then we have another sequence that produces a gap. Then we have the third gene, and after the third gene, we have the stop codon. Okay, so now, yeah, we are ready to say bye-bye. I hope you have a nice day. See you in the next session. In the next session, we are going to see how to edit these genes. So the real sequence, you are going to see the real sequence, okay? That is a series of copy pastes, okay, in the correct position, the proper location. The location will be the one that you have seen here, okay? We will follow this design. So and now, for real, have a nice day. See you in the next session. Bye bye. Have a great day.